To recognize and respond to the antigens that are their specific targets, both B cells and T cells carry special receptor molecules on their surface. For the B cell, this receptor is a prototype of the antibody the B cell is prepared to make, anchored in its surface. The T cell receptor, or TCR, is more complex. Structurally, it is somewhat similar to an antibody, made of a pair of linked chains with variable and constant regions. Unlike a B cell, however, a T cell does not seem to release its receptor and cannot recognize protein antigen in its natural state. The antigen must be first broken down and the fragments or peptides bound to an MHC molecule by an antigen-presenting cell such as a macrophage. Helper T cells, usually CD4+ cells, look at antigen bound to class II MHC molecules, a combination displayed by macrophages, B cells, dendritic cells, and some other cells. Cytotoxic T cells, usually CD8+ cells, on the other hand, respond to peptide bound to class I MHC molecules, which are found on all nucleated body cells, more importantly. The TCR thus forms a three-way complex with its specific foreign peptide antigen and MHC molecule. This complicated phenomenon is called MHC restriction, the limitation of T cell action by the presence of class I and class II MHC molecules on immune and non-immune cells. Like B cells, T cells express a clonal antigen-specific receptor. The TCR is similar to immunoglobulin. It has two paired polypeptide chains, both of which have constant and variable portions, and both of which are composed of immunoglobulin-like domains. Two important differences are: the TCR is monovalent, and it exists only as a cell surface receptor and has no counterpart to secreted antibody. TCR genes are similar to Ig genes in their organization. The TCR beta locus contains V, D, and J segments like the IgH locus, and the TCR alpha locus contains V and J segments like the Ig light chain loci. In fact, the mechanism of gene rearrangement for TCRs is essentially identical to Ig gene rearrangement. Mutations that lead to a failure of rearrangement affect both Ig and TCR. Such mutations produce a lack of both T and B cells, and therefore a complete lack of acquired immunity and a severe immunodeficiency that is fatal, called SCID, S-C-I-D. The combinatorial diversity of TCRs is similar to that of Ig. We're talking about their location and organization of DNA recombination, signal sequences, or RSSs of germline TCR DNA. However, there are a few major differences between TCR and Ig receptor diversity. 1. TCRs make more use of the N region, that's the non-template encoded sequences, than do Ig. While it occurs in all TCR chains, this only occurs in the Ig heavy chain portions. The P region, the repair of single-stranded ends of coding sequences, generates palindromes. This diversity for introduction of new amino acids in the CDR3 region. This makes sense because the CDR3 region interacts with the antigenic peptide in the MHC cleft. The CDR3 region is significantly more diverse than CDR1 and CDR2. 2. D segments can be read in all three frames. When the D beta 1 region of different T cells was analyzed, it showed that the D region could be translated into all three frames. For example, GGG, ACA, GGG, GGC. Say that's frame 1. GGA, CAG, GGG. Say that's frame 2. GG, GAC, AGG, AGG, C. Say that's frame 3. Let's go back and do frame 2 one more time. G, GGA, CAG, GGG, GC. These are all codes or codons. Typically in the Ig heavy chain locus, the random addition of N nucleotides and P nucleotides leads to recombinations that are out of frame in 2 out of 3 joinings, which leads to termination codons in immunoglobulin. This is not the case in the beta TCR. Its D segments do not contain nonsense codons in any of the 3 frames. This leads to the maximum number of rearrangements during DJ and VDJ joining. We'll get to that and those numbers in a moment. 3. D to D segment joining does not occur in immunoglobulin, but it does occur in TCR. Beta and gamma chains can have 4 V region joinings: VJ, VD, DD, and D to J. The RSS flanking of D segments is different. 6 N at the 4 junctions. Alpha and gamma chains do not have D segments. I will repeat, alpha and gamma chains do not have D segments in their germline DNA. 4. TCRs do not undergo somatic hypermutation. There is no mechanism for selection of any somatic mutations that might naturally occur in TCR genes and no affinity maturation like in immunoglobulins. There's more info to come, but make sure that you start over from the beginning of this section to read more or to hear more about the T cell receptor and its associated complexes.